Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today we will be learning about lambda phase vectors, and in this session you will be learning about bacteriophage, the lambda phage, how it is used as a vector in genetic engineering, and how the wild lambda phage they differ from the recombinant phages, the insertional vector and replacement vectors, its advantages and limitations. Dear learners, let's first study about the bacteriophages. These bacteriophages are the virus particles which attacks the bacterial cell. The study of the bacteriophages was initiated in the year 1940 by the scientist Max Delbruck. If you see the bacteriophages, generally they will be appearing as tadpole shaped, having a head and a tail, whereas some bacteriophages they will be having only head and some are filamentous. The genome can be either single stranded or double stranded, and the nucleic acid it may be either DNA or RNA. And the genome it is encapsulated in protein coat known as head. So here you are seeing the picture of a typical bacteriophage, that is the T4 phage, which infects the E. coli cell. So this particular bacteriophage, it is having a head, a tail and a tail fibers. The head is called as capsid or protein coat. In this protein coat, the genome it is genome is present, linear double-stranded genome it is present in this head, and by means of this tail fibers it will be attacking the bacterial cell and injecting its genome. Coming to the type of phages, the bacteriophages are divided into two types based on the mode of propagation. So it propagates either by lysogenic cycle or lytic cycle. In lysogenic cycle, when the bacteria is attacked by the virus, the viral genome enters into the bacterial cell and the genome it gets integrated into the bacteria main chromosome and it propagates along with the replication of the bacterial DNA, and this type of cycle is known as lysogenic cycle. The second type is lytic cycle. So when the bacteria infects this particular bacterial cell, the viral genome it remains independent and it takes over the host metabolic machinery and it propagates, multiplies. The viral DNA it gets multiplied into many copies and it also synthesizes the proteins, viral proteins, head proteins, tail proteins and other packaging proteins, and all of these this particular DNA and the proteins they get assembled to form complete viral particles in the bacterial cell, and afterwards these particles they break the bacterial cell wall, and that is the bacterial cell it gets lysed and the full-fledged viable viral particles are released outside. This type of cycle is called as lytic life cycle. So the temperate phases are the type of virus which can propagate either lytic cycle or lysogenic cycle. So it can be switching on to this type of either lytic or lysogenic mode depending upon the nutrients available and also the metabolic status of the bacterial cell. Whereas virulent phages they undergo only lytic cycle. In recombinant DNA technology, phages propagating through lytic mode are preferred as more number of DNA copies are produced. Further, the plaque formation exhibited by the lytic phases help in screening the transform cell. So in the picture which you are seeing, these plates they represent a lawn of bacterial cells and here and there you are able to see some clear zones. These zones, they represent the lysed bacterial cells. How these bacteria cells are lysed in this portion means due to the attack of the lytic phases. So these zones, they represent the viable lytic phases. So by means of this the plaque formation, you are able to screen the transform cell. So due to these reasons, the lytic phases are more preferred in gene cloning experiments. Next, coming to the actual example which you will be going to study, the lambda bacteriophage. Why this bacteriophage is selected for gene cloning means, particularly it is easy to transfect the bacterial cell and it is having the ability to accommodate large size DNA molecules. These lambda bacteriophages, they have linear double stranded DNA and the size is 48.5 kb with 50 genes. It is a temperate bacteriophage of the family Styloviridae and it uses E. coli K12 as its host cell. And this virion has a icosahedral head and a non-contractile tile, a base plate, tile fibers and binding proteins to bind the bacterial cell. Lambda phage genome is a linear double-stranded DNA having overhangs at its 5' ends. The size of the overhangs is 12 nucleotides and these bases are complementary in nature. When the linear genome enters into the bacterial cell, it gets circularized due to the base pairing at the overhangs to form a site called as cos site. So in this manner, the linear genome is becoming a circular genome in the bacterial cell. And if you see the components, different components present in the bacteriophage genome, it is having replication related genes, integration and assembly, phase assembly coding genes. So if you digest the phage genome into 3 parts, that is the first part and the last part, they contain essential gene, whereas the third middle fragment, they contain non-essential gene. Thus 2/3 of the lambda genome, it contains essential gene and 1/3 portion it contains non-essential genes. Approximately 24.6 kb of the lambda genome can be deleted and a maximum of 26 kb foreign genes can be inserted in a wild lambda phase. So during the construction of this lambda vector genome, the size limit is the main factor which determines the viability of the virus particle. So during packaging, that is the packaging of the DNA into the phage head, the size should be a minimum of 40 kb and a maximum of 53 kb. If the size is less than 40 kb or more than 53 kb, the lambda genome, it fails to get packed into the head of the virus. So thus the genome size, it restricts packaging and plaque formation, which helps in easy screening of the transform cells. Coming to the replication in lambda bacteriophage. This lambda bacteriophage, it replicates by rolling circle mechanism, and these cos sites which we have discussed, they will be having internal restriction sites, and the replication, it results in the formation of long contameric molecules. It is a polymer. So this contains several linear arranged phage genome units, that is monomeric units, each separated by cos sites. Thus just before that is the packaging of this DNA into the capsid, the contameric DNA is cleaved at each cos site and a single unit of this lambda DNA is packaged in each of the head. So this is what is represented here, the rolling circle mechanism, and during the replication you will be getting a long contameric DNA, a polymer which is having numerous phase genome units which are separated by like this numerous phase genome units. Each unit is separated by the cos site. Okay, that is about the rolling circle type mechanism. Thus you have seen the non-essential genes in the lambda genome is replaced by our desired gene to construct a recombinant DNA molecule. So these recombinant lambda phase genome, it can be assembled with the phase proteins by means of a method called as in vitro packaging. So during this process, the packaging proteins are produced by infecting the bacterial cells with mutant lambda strains. So when you infect the bacteria with these type of mutant strains, they are able to produce head proteins, tail proteins, and packaging proteins. You will be extracting the cell lysate, and this cell lysate is mixed with recombinant lambda DNA under in vitro conditions for self-assembly process, and at the end you will be seeing a complete viable recombinant lambda phases which are ready for gene cloning experiments. So that is what is represented in the picture. This is the long contameric DNA which is having a individual phase genome units separated by cos sites. These cos sites will be having recognition sites for restriction enzyme. So when you cut them with this contameric DNA is digested with the restriction enzyme, individual phase genome units are released and they get packed into, each head will be taking the lambda phase genome unit, and then the tail will be coming and attaching the head. With the help of this packaging proteins, you will be able to get a complete viable recombinant phase which is now ready for gene cloning experiments. Coming to the type of lambda vectors, you know that the wild lambda phases are not directly used in gene cloning experiments because it is containing a lot of non-essential genes. So by deleting these non-essential genes which are promoting the lysogenic cycle and retaining the essential genes. So the essential genes, they include those involved in replication, integration, and lytic cycle. So these essential genes, they are retained to create a recombinant lambda vector. The recombinant lambda vectors, they help in carrying large size DNA inserts up to the size of 25 kb. The recombinant lambda vectors which are used in gene cloning experiments, they are insertion vectors and replacement vectors. Coming to the first one, insertional vector. Here the foreign gene is inserted into the wild lambda phase genome by cutting the genome at one site, and this site is also present in the marker gene, that is LAGZ gene. So here you can insert a minimum of 4.3 kb and a maximum of 14 kb of the foreign DNA inside the insertional vectors. So these vectors, they are generally used for the construction of cDNA libraries and also for cloning the mRNA sequences. The examples are lambda ZAP and lambda GT vectors. So it is what is seen in this diagram. So it is having a single restriction site which is present in the marker gene, LAGZ marker gene. So once you are cutting that site and inserting our foreign DNA in this particular insertion vector. Okay, the second type is the replacement vector. So replacement vectors, why they are used means in order to overcome the limitation of the insertion vector. So in insertion vector there is very little scope to insert large pieces of DNA, but by using this replacement vector, the stuffer DNA, that is all the non-essential genes, they are removed and they are replaced with our foreign gene. So we have learned that nearly 24 to 25 kb of the lambda genome, it contains a non-essential gene. The entire thing, it is removed. That one is called as stuffer DNA, and that DNA is removed, and in that place you can add our desired gene. So here up to 10 to 23 kb of the foreign gene can be inserted in this replacement vectors, and these vectors, they are generally used for the construction of genomic libraries, and the examples are lambda EMBL and lambda EMBL 3, EMBL 4 and lambda dash, etc. The replacement vector, it will be looking like this. So this is the non-essential genes which are present in the wild lambda phase. So that portion, it is removed, and in that place our foreign gene, it is inserted. So on either side of this stuffer fragment, you will be having two restriction sites, that is on either side you will be having this restriction site, so that by means of the restriction enzyme digestion you will be removing the stuffer fragment and replacing it with our foreign gene. So in this manner you can create replacement vectors, and you can clone large size DNA inserts in this particular replacement vectors. The methods used for screening recombinant lambda vectors are plaque formation and blue-white screening. And you people, you know that the genome size limit restricts the packaging and viability of the virus particle. If a genome of correct size, it gets packed into the capsid, then only the virus is viable, it multiplies and it shows plaque formation. So the plaque here, it represents the regions where the virus particle is actively growing. The clear zones, they represent the plaque. In a non-recombinant virus, because the genome size is less, since the foreign DNA is not inserted, so it is unable to grow and multiply, and you are not seeing any type of plaques in non-recombinant virus. So plaque formation, it indicates the transformed cells or the recombinant lambda vectors. Coming to the second method, blue-white screening, and we have seen that in insertion vector, the foreign gene is inserted in the LAC-Z marker gene. So the transformed cells here, they appear white colonies, whereas non-transformed cells, they appear as blue colored colonies. In this manner you will be able to separate or select the transformed cells easily by means of these visual indicators. And at last, let's recap the main points. So you have learned that bacteriophages are nothing but the virus particles which attacks the bacterial cell, and in that different types of bacteriophages, you are taking lambda phase as because it is having many essential features of a vector. It can insert a foreign DNA up to 25 kb of the foreign DNA can be inserted, and they easily transfect the host cells by lytic cycle. And by removing the non-essential genes and by in vitro packaging, you are creating this recombinant lambda phases. So the two types of vectors which you have discussed are the insertion vector, and it can accommodate a maximum of 14 kb of foreign DNA insert, and they are used for the construction of these cDNA libraries. Whereas the replacement vectors, they can insert up to 23 kb of the foreign DNA, and they are generally used for the construction of genomic libraries. With this, I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.